Howdy, everybody. Thank you for joining today. So we're going to be doing a little bit of a different format here. I have a really incomplete and poorly prepared for a little small presentation of hop latent viroid that we're going to be improving upon, but we're going to talk through some of the latest research and topics of hop latent viroid. This has been something a lot of people have asked about, and I want to point you guys in some really good directions of resources, talk about some of the latest research, talk about what we do to prevent it, and then field some of those questions. So I'm going to go ahead and share my screen. Yeah, I need to really improve this, guys, but bear with me here. I'm going to talk through most of this. The first thing to talk about what the difference between a viroid and a virus is hop latent viroid is just a piece of RNA. So it doesn't have any other parts to its body, so to speak, like a virus does. And a virus usually has the protein sheath that allows it to penetrate the nucleus of a cell, which makes it about 1 million times more. Sorry, 1 million is not technical, but which makes it much more able to spread itself around in a plant or animal's cells. So it really it goes in. It's able to get through the cell membrane or cell wall, or hitchhike through into the cell, and then penetrate through the nucleus, and then hijack the equipment in the cell to make new DNA or RNA, and then replicate itself and go crazy, and therefore destroying the cell. That's what viruses do. Viroids don't have that capability. They literally just interfere with that, with those processes, but passively. They're not able to get into the nucleus themselves alone. So the viroid is just 256 nucleotides. Circular is not exactly right. They've actually found it's in the shape of a stick. It's in the shape of a stick shape. But it does have a shape where it goes up and around. I think I can visualize this for you guys soon. But this viroid cannot move around actively, and really it's physical transmission that is proliferating it. So that's going to be shears, physical touch, trays, pots, equipment, stuff like that. So cleaning has never been more important. We'll get to that. But it's really important to make the distinction that this thing is not alive. This is not a living thing. This is just a piece of RNA. It's like a spike strip on a road. It just can give you a flat tire, and you just have to avoid them. So let's keep going through. So Toomey Genomics. I've been turned on to this organization. They do some really great work, and they've done some really awesome research on this. And again, I want to direct you guys to some of the best researchers out there for this viroid right now. And Toomey's definitely at the top of their game. So check their website out. They have a lot of really good resources there. Okay, what does this viroid do? I think a lot of you guys are familiar with this at this point, but the symptoms are pretty what do you call it? They're subtle. It's not really, the plant doesn't catastrophically die. It just kind of withers away and becomes less and less effective. I think a lot of growers can have this experience, and you almost you think am I crazy? This plant used to be really vigorous, right? Because it's over time that it kind of declines. But then at some point, when it does become obvious, it's often way too late. So the things you'll see with the virulence of this are often lack of proper bud formation and yield. Hop latent viroid positive plants never have dense, nicely formed buds. They're always kind of airy or fluffy, or they look like they weren't grown properly. The testing results is obviously doesn't have the potency or terp. It has a flat smell to it. Propagation is oftentimes interrupted with this, where you really have trouble cloning the plant, and you don't get a vigorous clone mother or plant structure out of it. leaf discoloration, oftentimes confused with some sort of deficiency. And in fact, it could be a deficiency, but it's, the viroid kind of influencing that. And then one of the telltale signs for me that I see is secondary branches. Instead of being like kind of having that nice like angle, they oftentimes have like a half pipe angle. Like they have almost a really like parabolic shape to them. That so that kind of looks like a candelabra. That is actually a really distinct symptom that I see oftentimes. So this was called dudding. Dark Heart really did a lot of research on this and found this. Anyway, we understand the virulence. Sorry, this is again, I need to really work on this. Transmission of this. This is mostly mechanical. This is where we just need to, our biggest thing is using gloves, washing clean, replacing our shears, and using proper disinfectants for our shears specifically, as well as not like reusing pots or soil or stuff like that, and spreading this viroid. Seed transmission has also been documented just recently. And one thing that's been talked about is it's difficult to know if the seed transmission originates from the outside of the seed or actually inside the seed. So that's something that I think there's more research. I'd be curious if you guys have any insight to that. But the actual so if you can imagine, if you plant a seed, you might say, oh my God, the seed had it. It's like it could be actually on the surface of the seed and then spread to the plant through the germination process. So that's one reason we always recommend, going back to the seed germination protocols on our website, to use a disinfectant such as hydrogen peroxide to clean the seeds before you pop them. And if you do put them in a solution, being able to shake them and really agitating that solution, because seeds have a really textured surface, and they're hydrophobic. That's why oftentimes even good seeds will float on the surface of water, and they'll hold some of the air against it. And you can imagine if you have a disinfecting agent, if the liquid isn't getting to it, it's not cleaning it. So there's also studies that show that this shape of RNA is a lot more stable than your average RNA. So it can last quite a while on surfaces. It can last 7 months in water, and it can last many months or years on surfaces. I'll give you guys next week this presentation with some of these citations on here. But yeah, this thing's a lot more stable than you think, which is pretty crazy. So think twice about using that old, 1 gallon pot that's clean enough if you've had an outbreak of this, because it could be sitting on there for like a long time. Okay, concentrations in sinks. This is an interesting phenomenon for viruses and viroids that they tend to collect in one part of the plant. And for virologists, this is kind of common knowledge that there's typically a best place to test the plant, and it has like all the viroid like collects there, right? And so again, Toomey Genomic did a study that found that the roots actually had the highest concentration of viroid, which I thought was very interesting. For our test, we test petioles. As petioles, again, petioles are a sink for the plant in a lot of ways. It kind of collects a lot of sugars and deposits stuff into the petioles. And it's also a kind of a exchange station between the leaf and all that metabolism and the stem. So so we like to test petioles as well. But it's very interesting to think about concentration when you guys are sampling. You should be aware that not all parts of the plant are created equal in having your test come out, like having a valid test, so to speak. Okay, testing. The only way right now to test for hopping viroid is through, that's not true. There's actually LAMP assays too. But the most reliable way right now to test for hop polyton viroid is our PCR assay. This is the same exact technology that's used to test for COVID-19. So I'm sure you guys are all like familiar with this. But what it does is it uses polymerase chain reaction, that's what PCR is, to amplify the DNA by using it actually changes the temperature in a bath. So it's like a hot water bath, kind of like a sous vide or something. And what it does is it changes the temperature and then goes back down, and it goes back down, and it goes up and down and up and down. And what that does is allows the essentially assembles DNA or RNA, and then puts it back down to the temperature that it can collect all the nucleotides and then reassembles it again and again and again. So it like copies them. So the number of cycles that it takes to then detect the actual RNA is referred to as cycle threshold. This is a really important number because kind of like golf, like the lower, not the lower the better, but like lower the number here, the fewer cycle threshold it took, meaning the more RNA is in the sample. So what we found, somewhat anecdotally, but with a lot of our laboratory research, is that if you have a cycle threshold of 10 to 15, this is catastrophic. The plant is not coming back. It's likely highly, highly symptomatic, and we're in big trouble here. 15 to 20 is severe. You probably have highly symptomatic plants, and again, your likelihood of coming back is very low. 21 to 25, you probably won't have. You might have inklings of symptoms here. You might have like here and there, but the plant might be performing like just fine. However, you still might have trouble remediating this. 26 to 30, you probably don't have any symptoms. And yeah, we can probably remediate the plant or reduce the amount of cycle threshold of the plant, and maybe it can get better. 30 plus, this is like barely a detection, and in our experience, it's likely a false positive, or it increased the likelihood of a false positive by a lot if it's above 30 cycle threshold. Okay, prevention of hop latent viroid. What the most important thing is cleaning of your shears, especially. Your shears are the best place to physically transmit it. Remember how we talked about how the viroid can't get through the parts of the cell? It can't transit through the cell like normal viruses. It relies on you breaking the cell, so having like a rupture in plant cells for it to get into the cells for the most part. So you can imagine if it's on the shears and it cuts through the cell wall, that's the best opportunity for that viroid to get in there. So cleaning the shears is critical. There's a lot of different ways to do this, but one thing I want to just sing from the mountaintops is isopropyl alcohol will not kill this viroid. Alcohol encapsulates the circular viroids because of just the chemistry of it is not sufficient to lyse or to break apart the viroid. So you need to use some of the stronger oxidizing agents, such as Virkon Est, Lysol. When I put Lysol, there's bleach products, but dimethyl benzyl ammonium chloride, active ingredient Lysol, and then concentrated bleach, probably between 10 and 20%, will kill the viroid, and any one of these is reliable. Bleach will rust your shears and degrade the shears faster than usual. So I prefer the Lysol because the shears can sit in those overnight and they don't really care. It doesn't like rust them. So that's a really important thing. And then like quarantines and moving in the way that your facility flows is important. For instance, having a staging area of dirty pots from harvested plants right next to your propagation area, it's not a good idea. These are some of the principles of like if you ever work in a restaurant, like some of the, hey, don't put your raw, chicken in a walk-in next to the dirty dishes. Don't put the cooked food next to the raw food. Don't put the clean stuff next to the dirty stuff, right? Allow kind of a process flow so that the dirty stuff comes in, or the clean stuff comes in one entrance and it cycles through and then goes out another entrance. this goes all the way to facility design and process design, as well as just like protocols and discipline of your team. Of course, if you really want to prevent hop latent viroid, you should get plant tissue cultured stock. So working with us to get bare pulse stock, or to get stock directly from tissue culture, is an amazing way to get kind of verified hop latent free. And some of the better nurseries out there, such as Cultivated Nursery, does excellent work to prevent viroid propagation within the facility by having, they have a dedicated shear for every clone mother. So they're really going above and beyond with that. And they're replaced all the time, says Sergio. Okay, so sampling for hop latent viroid. This is the last kind of thing that I think probably causes a lot of confusion for people, which is that proper sampling for this is important. So if you take samples, let's just say I take a sample of, I want to see if this clone mother has hop latent viroid. So I cut a leaf off and put it in a bag and send it to the lab. It could come back negative because the viroid is patchy. Like we talked about, it can't like penetrate through. An analogy that could help imagine this could be like a skin cancer, or cancerous mole on your arm, right? If you sample a piece of, part of your leg, it's going to say, oh no, you don't have skin cancer. And it's like, well, you have to sample the part that has it, right? So what we want to do is sample the plant in a lot of different places and then compile those samples together. However, if you present a laboratory with a sample, here's a bag of 50 leaves, they're not going to necessarily go ahead and compile all those for you, because their machines can't extract DNA from that big of a jumbled. It's got to fit in like a test tube, guys. So if you could imagine compiling the samples it has to fit into like a shot glass for your to put a visual example. So one thing that we do is we take a combined sample of like petioles, for instance, cut a bunch of leaves off. Let's say you take 10 leaves and you get all the petioles. We then line them up and cut the ends off all of them, and then compile those ends into a test tube and then sample that. And that way we know that the whole, a smattering of the whole plant got sampled, so we can have a really accurate yes or no. Obviously that could dilute the sample in a way that one branch has a 15 cycle threshold, another branch has a 30 cycle threshold. But really what you want is a yes or a no. Is this plant sick? So compiling the samples is one way to prevent that. I can again make I'm going to be working on this a little more. So I can make a better sampling protocol for you guys that you can actually print out and put on the wall in your grow for your technicians to follow, which can help you get more accurate sampling. Okay, cool. So that's pretty much my mini presentation for the day. I'll improve that obviously, guys, but I think we'll just take it away with the questions, and I'll let Sergio and Anat and Christian here kind of go to town with selecting questions and leading the conversation. So thank you, guys.
Of course, yes. Thank you, Chris.
Chris, can you go over a little bit about the peroxide protocol for seed germination and why it's important?
Yeah, so we talked about the potential viroid on the surface, but viroid isn't the only thing. Viroid isn't the only thing that is present on that. You can also get a lot of molds and funguses on the surface of seeds because the seed cleaning and being able to process the seeds and mature them properly, you usually kind of like pulverize the seed stuff into like dust. So I highly recommend cleaning the seeds, and hydrogen peroxide is I do think gosh, I need to check to verify that. I see a comment here, but I'm pretty sure hydrogen peroxide is also capable of killing the viroid. It's a strong oxidizing agent, whereas isopropyl alcohol is not. It is semi-polar and soluble in water. So it's a little bit of a different thing. So yeah. But let me, Det Chronic King, let me check on that again. I'm pretty sure it does kill the viroid. Thank you for that question. I see Norcal asking about gibberellic acid in the germination protocol. I don't think that's necessary, and gibberellins, guys, are serious hormones. So they really shouldn't be trifled with as far as like, we're talking micromoles of concentration make an enormous difference. So like 10, like, so that's I think that's a micromoles for that would be like if you have a liquid solution adding 5 micrograms to a gram of solution to a liter of solution, which is like a tiny amount. So it definitely makes a big impact, and if you overdo it, it will elongate the seeds and etiolate the seeds. Etiolate's a good word here, to the point that they'll fall over and die. Yeah, it's really that can be really catastrophic. So, thank you for the comment, Det Chronic King. We'll look into that too. We do viroid screen all of our seeds, but in general, you should wash them. So I'm gonna look that up too. Thank you, Det Chronic King. But yeah, gibberellic acid, I wouldn't do it just because I think there's probably more risk there than benefit, even though it has been proven to increase like germination rate and success of seedlings. I think we have a different channel for the actual questions, correct?
We do. There's a question from Happy Dudes Road. Some guy told me the big plants equal low potency. What's your opinion?
Oh no, I don't think that's the case. It's an interesting. There's kind of something there, but not really, because if you have obviously the potency measurement is weight, and mass divided by how much by the assays they do to determine concentration of cannabis, right? Sorry, concentration of THC. So if you do have a plant that has more water weight or has bulky parts such as stem and everything like that, then like you guys have probably done this. If you submit a COA sample that's like untrimmed or has stem and stuff, it's gonna come out lower potency. But big plants low potency? No, I don't think that's the case. I've seen some really big plants that test really, really high, and some little plants that test low. So it's most genetics, light intensity, nutrients, success of your grow, flowering time. That's I think probably the most important factors for potency.
Okay, we have a question from Organic Greens 510. What does the ideal mother plant look like when taking clones or keeping it for clones?
That's a great question. The ideal mother plant should have as many. So it should have. I should get an illustration here, but it should have as many mains going up as possible, and should be either like a flat, like a broom shape, so like an inverted broom shape, like that. But it could also be, it's going to naturally have kind of a diamond shape. So if you train it out like this and just let it grow, it's going to create a diamond shape where the very top in the middle is going to be the tallest one and the sides will be a little bit stunted, right? So the optimal shape when you're actually training it should be a broom. So it should be like a triangle like that and then flat on the top. And training people to do clone mother management, I always suggest the inverse triangle technique, in that if you have that diamond shape, like that natural shaped plant, what you're going to do is cut the very middle, like the top apex in the middle. You're going to prune that really hard. And then like one layer out from that, you're going to prune that like kind of hard. And then the actual outers, those outer branches, you might not prune at all or prune very lightly. If you do that, then you create a little bit more of a that broom shape and that even shape, because you really don't want that like apical dominance. But it also highly depends on the genetics of the plant. We've seen that there's 3 different general morphologies of cannabis plants. You have the highly apical dominant plants that just grow straight up like a cane. You have the ones that are kind of like natural, like average, that grow kind of like a diamond, like that. And then you have the very bushy ones that really like to go out and kind of stay like low and out. So the low and out ones, you barely need to train those at all. You kind of just cut the tops. The average ones, you need to put that inverse triangle. And the apical dominant ones, you really need to prune it down hard. So, yeah. And leafing and bottom pruning your clone mothers is extremely important.
Okay, thanks. Dushi asks, how can I ask for more information about high society strain and possibly a double stack? I already asked, but maybe you forgot. I'm wondering what generation it is, F1, F2, or S1.
Okay. High society, both high society and double stack are F1 generation seeds. So we have high society is Biscotti by Jet Fuel Gelato. So yes, that's a single generation F1 seed. And then we have Apple Fritter by Jet Fuel Gelato for double stack, which is also F1 seed. Anat, where was that? What other questions were there?
That was it. It was about, is it enough? Yeah, that's the whole question.
Okay.
Moving along, this is from T. Adams. I currently use general hydroponics, and I've been wondering if there's any other hydroponic nutrients that are worth buying.
The GH is a great company. Every hydroponics nutrient solution has its like benefits and drawbacks, and I think the main compromise that exists with the hydroponic solutions is ease of use, pretty much, right? So the 2-part solutions are really reliable. There's much less like room for error for you to screw up such a simple recipe. But they might be leaving something to be desired for some of the chemistry that's possible with a 2-part solution. There's a reason those 2 parts are separated when they're salts. You're preventing reactions that only happen in a solution. So the multi-part solutions, I think GH has a couple additional parts, have potential benefit, but it's just a little bit harder to work. Generally, I would strongly recommend using salts, because any of the liquid nutrients, you're paying a lot of money for water. If you actually do the math on the salts, you're mostly like for them to dilute it for you, essentially, you're losing a lot of money. So that would be my recommendation. Use salts, not liquid. And GH, a lot of the different brands. GH is a great brand for hydroponics. So yeah.
Question is from SMP Mang. Does Compound offer regular seeds?
That's a really good question. We've worked almost exclusively with feminized seed, and it's kind of become our thing, mostly because we stabilize these really amazing clones, and then we want to reverse those clones. So, no, not really, but all it will take is a male from a line that we really like to kind of kickstart that. And I'll give you guys a sneak peek for one of those things, which is some cherry lime dog seeds I have from BioVortex. If you guys are familiar with Jesse of BioVortex, is a good friend. And we're going to be hunting those, and I'm sort of interested in saying, let's try to get a male from that. There isn't any proof yet that there's unique or valuable genetics that are only transferred by males and not reversed females. But I think, I'm willing to be proven wrong in that. And I think the more we discover about the genome, we'll probably see that there's certain things that only transmit with that, or reasons why going to regular seeds periodically are better. So I think that's a really good question. I think our radar is open for that, and I think you will see some regular seeds from compound genetics at some point, but we really kind of become known for FEMs, so we'll see.
Next question is from Organic Green 510 again. How can you keep a mother plant around for a long time?
It's challenging. Avoid stress, avoid insects, reduce stress as much as possible, like saying, pruning it on time. One of the most difficult things that happens is once you get a really big plant in a pot, it can get root bound, or it can like collect too much salts. So you want to keep it like the right size for the right pot, which just means you need discipline to prune it back. And that's one thing, running a nursery and taking care of a lot of clone mothers. Even if you don't want the clones, keep on a clone schedule anyway, and just cut the clones and put them in the trash. But you can't let that thing kind of just like go crazy. And I think it's a little bit of a misconception that you should just let it, it'll be healthier if you just let it go. It's like, no, cut it. Keep it the right size. Screen it for viroids and treat it with beneficial insects, treat it with pesticides. And if you do have any inkling that the plant's unhealthy, take a clone and restart a tray of clones from that clone mother. If you take 10 clones from the clone mother, only grow out the best 1 or 2 clones from that tray and discard the rest.
Well, this is a good segue to the next question. How many times can you clone a clone before it starts regressing in development?
Oh, this is a layup question for my favorite thing to talk about. So there's a phenomenon called bioaccumulation. It's usually that I kind of like talk about with clone mothers. It's usually reserved for trophism or biotropism, and the way that like eagles get a lot of poisoning in their bloodstreams because these food chain like concentrates all these pathogens and everything and the contaminants into them. But the clone mothers, these plants will every scar, the plant's not like gonna kind of like take that in. It's gonna get scarred from it. It's gonna get a black eye from those things, right? So I've always said like you can look at a grower's like flowering rooms and be like, oh, isn't it amazing? And I'll be like, let me see your clone mothers. And if the clone mothers, I can kind of read those and go like, you had some issues 4 months ago, and they, oh my God, the whole place, so like those clone mothers will really take that in. So some people are really good at that and can hold it for a long time if the stress levels are low. But all it takes is that AC going out, the insect infestation, the viroid thing, the fusarium outbreak because the heater on the chiller on the hydroponic system, went out, something like that. And there you go. Like those moms aren't good again. So protecting them to have them in 2 stages, clone mother and small clone at all time. That's one way you can do that. But tissue culture is really the only way to prevent that long term. And this is really why you don't see a lot of classic strains out and about. The clone mothers were too difficult to maintain.
Okay, we have a question from Portkey Cannabis. What do you do for the final 2 weeks of flower and for harvest? Also dry and cure.
Oh, that's a big question. I'll answer the last 2 weeks, but dry and cure is that's a whole nother can of worms. But the last 2 weeks, you should be looking at reducing EC in your root zone. So what you want to do is mimic the fall, essentially, and what the process that you're trying to kickstart is called senescence. I'm going to put that in the chat just because it's a good word, one of my favorite words. So it's it's essentially the ripening of the plant, and plants have a programmed cell death, that it starts to like say, oh gosh, it's getting about that time, isn't it? And it starts to break down its own like tissues, and essentially dedicate that as a last ditch attempt at maturing its seeds and just dying. So what you want to do is reduce the EC in the root zone by reducing the amount, either flushing with 0 EC or reducing the EC of the feed, reducing temperatures and reducing humidities. And you can also run a higher VPD in those last 2 weeks than you can for the rest of the run. So that's what you're looking at in those last days. And obviously drying and curing, you want to do that low and slow, around 60 degrees Fahrenheit, 60 degrees, 60% humidity. A lot of people will disagree with those estimates, but no one is more than 10 degrees off that, 10 degrees and 10% off that general setting.
We have a question from Gilberto. What new compound genetics flowers are we expecting to come out as a flower line?
Oh, that's a great question. Maybe Sergio can help me out with that. New flowers coming out with compound flower line.
Sergio, okay. We can't hear you.
I'll repeat this. So we got Goofy's Pheno that we really like. So that's Apples and Bananas by Jokers. That's coming down the pipeline. We have Rose Gold Pave. Rose Gold Pave was from the Pave line and is just so much gas, guys. Like this is really going to represent one of the like the gassier strains, and which wasn't as well represented in the previous 5 lineup. What else we got, Sergio? Okay. So we're working on a pheno of persimmon to come through, which is a Rainbow Cheddar by Gastropop. And again, we love those like Z terps and some of the crazy expressions with the cheese and Gastropop. So we're really trying to do something unique there. That's kind of our X Factor one.
Okay. Pinkman 4i20 asks, would there be any collaborations with banks like Soulfire, Exotic Genetics, in-house being worked on?
I'd love to. We've worked with a little bit of the red pop from Exotic Genetics, but I'd love to do a more formal collaboration with them. In-house is great. I love their Platinum Kush Breath is one of my favorites of all time. And I really like a lot of their gear. I also really like Swamp Boys and would love to work with them. I don't know that much Soulfire gear, but yeah, I mean, we're always doing collaborations. So, so much of this is sort of serendipity. We see them at the big expos, we see them out and about, and then we just go like, wouldn't this be fire? Let's talk. So I think we just got to run into these guys and talk. But I respect all of their work, and then the red pop, which we've worked a little bit with some of the couple lines here. I'd love to work with these guys.
Fire Gardens Farm Gen is asking, you guys going to ever release the Legend Orange Apricot again?
We have it still. Yeah, I mean, so one thing, I've kind of talked about this, I've had a stump speech about this a couple times, but terpinolene is, the Legend Orange Apricot crosses typically have terpinolene as the terpene, which, is colloquially known as like the Jack terp. And I love that terp. I think there's a lot of really cool crosses, and I think it's a little reductionist, or like anything that has that tastes like Jack. But it kind of has a little bit of like you can't sell packs that have that terp. However, we have some standout crosses like Blue Apricot, Glazed Apricot Gelato, Mafia Funeral, tons of gear from that line that we absolutely adore, and we're holding them in our bank and going, wait for the right moment. So we'd love to work that line again, and I really appreciate that question. Yeah, I love some of the gear coming out of that. We still have the plant, and I'd love to do a re-release of some of those crosses. That'd be really awesome.
NorCal is asking, one second. The screen just moved. Norcal is asking, are you guys implementing any gibberellic acid in your germination protocol?
I think we already addressed that question. Not.
Yeah.
No, we are not.
Okay. And one more question. If you have time, when you had the viroid levels at the catastrophic, does that mean it can't be remediated through TC, or it's just not worth the time?
That's a great question. In our experience, it cannot be remediated through TC. Essentially, what you're doing with TC is cutting out a small piece of the plant and hoping that that dissection does not have any viroid, which sort of depends on the assumption that some parts of the plant don't have the viroid. If you're at cycle threshold, or sorry, what cycle threshold was he asking about?
Let me go back to the question. The viroid catastrophic, does that mean you can't remediate it?
Okay, at the catastrophic, correct. In our experience, you can't, but it starts to get to kind of a diminishing return situation where it takes the two ingredients to remediate are time, money, and tests. So, man, we worked one line for almost 2 years and still couldn't get it clean. And when we look back at how much money we spent on viroid assays, how much time we spent on this, and then how many resources and money we spent to actually work the line, we started to say man, this strain just isn't really worth it. I mean, we could have bred a new seed line and hunted it in that period of time, right? So in our experience, it's just not worth it. But it might be possible. It's just extremely difficult.
That concludes all the questions that were put on the chat. Anything else you want to close with, or is anyone? Oh, I see a hand coming up. One more question. When you say catastrophic, that plant must look awful, correct?
Correct. Yes. I assure you, if you get a plant that tests at 10 or 11 cycle threshold, it means that there's these viroids are tiny, and usually they're in pretty small quantity. We're talking the plant's chock full of them to hit at 11 cycle threshold. To give that context, guys, again, the COVID-19 is the assay for it is almost identical. It's very, very similar. Same equipment, very, very similar, right? It would be common to catch COVID-19 at a 20 or 25 cycle threshold. Oh, you're positive you have COVID. So, if you're going to catch a virus or viroid through PCR assay and it hits on cycle 11, you have a lot of that in your sample. Good time.
We have one more question from Crimson. What terpenes in the persimmon and the wing sweet give it the butterscotch flavor? What combination of
that's a good question. I don't know. I think almost everything or most things in the gastropop seed line are myrcene dominant, but I don't know. I mean, one of the things, Crimson, that's important to put out there is terpenes are. You can have two flowers with the exact same terp profile on your COA. And one, everyone swears to God smells like banana, and the other one is gassy. Aroma is not necessarily indicative of that, even though they tend to group together. So I really don't know what that combination is. I have no idea. I'd love to know, but it could be something like a flavonoid that is not necessarily a terpene. And again, the testing labs to actually determine what things actually smell like need to test a heck of a lot more than just terpenes to determine that. So I'd love to be able to learn that scientifically, but I really don't know. I don't know.
And the last question is, what did we do to win the pack of seeds for tonight's show? I'll answer that. we're going to look at all the comments, and we're going to contact one of you guys, and you will be the lucky winner of the seed pack for tonight. Okay, do we have any other questions out there? Oh, here's one. Does hop latent viroids spread in soil?
I think it can be spread in soil, but I wouldn't say that it actively does, because also the bioactivity of soil might be less likely of it to be doing its thing. So that's a tough one. I think because of its physical contact, it could spread in soil, but I wouldn't say that that's a active or a primary way that it vectors. I really wouldn't. I really wouldn't know about that. Oh, I see a question about Tokyo Snow. Hibachi Supreme is one of my is really cool clone. It is definitely not a Japanese land race. It's from another breeder, but it's a Kobe. Oh gosh, what the heck is it called? It's a Kobe cross. It should be online, but it's one of the most unique plant morphologies I've ever seen. As most luxurious and waxy leaves I've ever seen. And it's a Kobe by, gosh, I can't remember. A seed finder might have it, but I'll put it in the chat later, guys, if I find it. When cloning, does a plant lose the original THC content from the mother plant? This is a Vern 671. Absolutely not. The clone does not lose THC content from the mother plant. And cloning off the mother plant to create a more uniform canopy is absolutely the best way to create high THC, high quality cannabis. As far as right now, I'm sure I'll have a lot of seed growers disagree with me, who, especially our clients and customers here, who grow amazing weed, intense. But as far as if you have a big canopy, clone has got to be the best way to do that, to prevent the different stretch amounts and different morphologies. I want to give you guys put a little bit of a shameless plug out there to the Whipple Effect book, so the Cannabis Cultivation Instructions book. I've rendered the recent, and I bet a lot of you guys were in attendance, the recent pheno hunting seminar I did and presentation I did into a chapter of a book. I'm really proud of it. Really excited to share it with you guys. And the Cannabis Cultivation Instruction, when we have the link there, we'll be sharing that with you guys. And I really hope you guys check it out and check out the chapter I guest wrote for the Whipple Effect Cannabis Cultivation Instruction book. So thank you guys.
I think this was a really great session. Thank you, Chris, for all your knowledge and for explaining things in a way that's very palatable.
Thank you guys. Thank you, Anat.
Yes, thanks, Chris.
I'll definitely follow up with this hop latent viroid thing and give you guys more information on that. And apologies for that being kind of a shoddy presentation there, but we'll improve it over time, just like we do with all these other things. So stay tuned, and we'll be sharing that with you guys.
Before you go, there's a couple of questions that came. What is the gaseous strain you guys have?
Oh, okay. A lot of the Pave line is extremely gassy, and a lot of the apples and bananas line is extremely gassy too. So, like we talked about, the rose gold Pave that we're putting in the compound flower brand was one that we are all just it's way on the side of just gas. And a lot of the crosses that are made with Pave, they sometimes still have kind of the mint from the menthol, but they have a ton of gas on that cross as well. Apples and bananas crosses, I'm trying to think of the ones that are gassiest from that line, but I'd say most things from the Pave line, rose gold Pave being one of those. I'm gonna look at the thing really quick. Give me 1 second, guys, because I want to answer this question properly. Okay, Facade from the apples and bananas line is extremely gassy. Rose gold Pave is extremely gassy. And then Honeycomb pave is ridiculous gas, pave S1. Uh, Candy pave with eye candy by pave. And rainbow pave. Those are all probably like the gassy collections we have right now.
One more difficult question. What is your favorite cross plant off the new gastropop drop?
Okay. It's got to be Tiger's Eye, guys. I love this plant. So many of these selections of this were just stellar, absolutely beautiful. God, all of them are really great, though. I really like 24 carats too, and Bling Blau too. Shit, actually, all of these are really great. But Tiger's Eye is the one. I really pushed that in Tiger's Eye name because I was just like, this is gonna be a classic, and we just knew it the second we saw it. So I would have to say Tiger's Eye is the one that is the apple of my eye.
Well, thank you so much. We're going to wrap this up. I don't see any other questions coming in. Thank you. And we'll see you guys next month. Oh, is there another question?
Thank you guys so much. No, that's good. That's good.
Okay. So we'll see you guys next month on the third Wednesday of the month. Thank you, Chris. Thank you, Sergio.
Thank you, guys.
Thanks, Chris. Everyone have a great night.